from Chulalongkorn Graduate Institute in Bangkok, Thailand. She obtained her doctorate degree in environmental engineering from University College London and resumed with her service in IMR upon completion. Her research interests are in the area of environmental health, environmental microbiology, and environmental engineering. For today, Dr. Asrah will be sharing her view on today's theme in her presentation entitled Understanding Antimicrobial Resistance from the Environment to Water to People. Asrah, the session is yours.
Bismillahirrahmanirrahim. Assalamualaikum and a very good morning to all participants. Thank you, Sophia, for the kind introduction. I am Asrah, and today I would like to share with you our research at the Health Risk Assessment Unit in my presentation today entitled Understanding Antimicrobial Resistance from the Environment to Water to People. My talk will be divided into 5 subtopics. In the first part, I will briefly talk about what is AMR and why understanding AMR in the environment is important. In part 2, we will look at the gaps and needs in terms of AMR studies in the water environment. In part 3 and 4, I will share with you the research being conducted in our unit, and finally, the summary. What is AMR and why it is important for us to understand it? AMR stands for antimicrobial resistance, which means any agent which is intended to inhibit or kill certain microbes but fails to do so due to the ability of the microbes that can resist the action of that particular antimicrobial agent. One of the examples of antimicrobial agents is antibiotics, the drug used to fight against bacterial infections in humans and also animals. The resistance occurs naturally when the administered antibiotics did not kill all the bacterial population, and the surviving resistant bacteria become stronger and expand to form a new population. The problem with antibiotic-resistant infection is it can be difficult to treat and even can cause death. As reported by the WHO, more than 500,000 people died every year due to drug resistance. The environment plays an important role in the transmission of AMR. As you can see from the diagram, the resistant microorganism can come from various sources, such as water discharge that flows from the community, hospitals, and pharmaceutical industry, before being channeled to the wastewater treatment plant and finally released into the river. The river water will be used for recreational activities, surface water source, and also as drinking water source. Although wastewater treatment plant treats the heavily contaminated wastewater, studies have shown detectable resistant bacteria in the river. According to the tri-cycle protocol developed by the WHO, both effluent water and river water are the two components to be monitored for the dissemination of AMR in the environment. As of today, there is no standard parameter for monitoring of AMR in effluent and drinking water standards in Malaysia, and there is no safe level established for AMR. The hospital's effluent measures BOD and total coliforms following the National Water Quality Standard for Malaysia and Environmental Quality Act. In the drinking water quality standard, the recommended raw water and drinking water currently measure total coliform, E. coli, and Clostridium perfringens to indicate fecal contamination in the water system. Both hospital effluent and drinking water system are currently being monitored by the Engineering Services Division, Ministry of Health, Malaysia. Therefore, further actions or key measures can be done by providing evidence to the stakeholders to reduce or eliminate the contaminants from entering our drinking water system. Hospitals are the major user of antibiotics, which contribute to their release into the environment. In the current study, we determined the prevalence of antibiotics-resistant pathogenic bacteria by testing them against 7 types of the most used antibiotics in the hospitals. This includes 2 antibiotics, considered as the last resort antibiotics, which are colistin and vancomycin. Samples were taken from the final effluent from the sewage treatment plant, or STP, from each hospital before being released into the wastewater treatment plant system. The effluent sample was transported back to the lab for immediate processing. Since the sample is heavily contaminated, the samples needs to be diluted to obtain a single colony for further analysis. Some preliminary findings from 2 hospitals marked as Hospital A and B. As shown in the bar graph, both hospitals recorded 80 to 100% of VRE and 10% ciprofloxacin-resistant bacteria in the Hospital A, and no resistance was recorded in Hospital B. VRE plays an important role in the transmission of hospital-associated infection due to contaminated medical devices causing infections such as in the bloodstream and UTI. For E. faecalis, 90% of vancomycin resistance were recorded in both hospitals, and no ciprofloxacin resistance was recorded. Presence of E. faecalis indicates poor hygiene and causes risk of infection due to immune suppression or contaminated medical devices. A drinking water system can be a reservoir for resistant bacteria, and we use water in our daily lives, which may increase the risk of exposure to AMR. In this study, we aimed to determine the presence of antibiotic-resistant bacteria, or ARB, in the drinking water system from the river water source, marked as 1, and the final point at the distribution tap to consumers, marked as 2. We measured the heterotrophic plate count, or HPC, grown on 7 antibiotic selective media of R2A agar. Lower concentration water samples were filtered using membrane filtration method, and samples were diluted for high load bacteria. Resistance rates or prevalence were recorded, and isolates were further analyzed for antibiotic resistance tests. The presence of ARBs before treatment and final distribution tab were compared at the drinking water treatment plant DWTP1. Resistant HPC was obtained in DWTP at the river source against all antibiotics tested. On the other hand, the DWTP has shown 0 number of HPC and ARB in the final tab, despite of high turbidity and bacterial load of the river water source. We have also observed presence of antibiotic-resistant Enterobacteriaceae from the river water source. Both Salmonella and E. coli were present at almost the same load, with 42% of them are multi-drug resistant, or MDR. More than half of the MDR were resistant to at least 3 antibiotics, and one isolate was resistant to all 7 antibiotics. Other studies have documented the presence of ARBs in drinking water sources as well as in treated water. China and Iran found resistant E. coli in water sources that were resistant to more than 3 antibiotics. ARBs were found in the chlorinated tap water system in the UK and the US. Similarly, this study found multi-drug resistant E. coli as well as resistant HPC in the drinking water sources. As a take-home message, the water environment plays a significant role in the transmission of AMR, which highlighted the importance of efficient effluent treatment from the AMR hotspots before being discharged into our rivers, and the importance of efficient drinking water treatment for human consumption. To end my talk, the control of AMR starts with us by using antibiotics only when necessary and disposing of the unused drugs properly. These are my references. Thank you, and back to the moderator.
Thank you, Dr. Asrah, for a very enlightening presentation. To all the viewers, if there are any questions that you would like to ask about her presentation, kindly submit your questions using the link provided in the YouTube channel box panel. Next, I would like to introduce our second speaker for today, Dr. Sakshaleni Rajendiran. Dr. Sakshaleni Rajendiran is a medical officer from Environmental Health Research Center, Institute for Medical Research, NIH. She received her Bachelor in Medicine and Bachelor in Surgery degree from AIMS University, Malaysia in year 2012. Following graduation, she did internship for 2 years in Hospital Seri Manjung and continued her service as a medical officer in Hospital Teluk Intan for 4 years. Then she transferred to the present institution in 2019 and currently doing Masters in Public Health in University of Malaya. She is interested in microbes, antimicrobial resistance, bioinformatics, and environmental health. Today we have Dr. Saksha with us to share her presentation entitled Antimicrobial Resistance AMR Through Environmental Lens, One Health Implication and Priority. Over to you, Dr. Sasha.
Thank you, Ms. Sophia, and good morning to everybody. So let's dive into antimicrobial resistant AMR through environmental lens, public health implication and priority. So here's my outline for today. Introduction, AMR in poultry environment, points to ponder, gaps and limitation, current project as in AMR in dairy farms. Okay, as we are aware, antimicrobial resistance is a global public health concern. Antibiotics, which have transformed medicine to a new exploration, was taken for granted. The wide usage not only in clinical settings, veterinary medicine, but in food production industry as well accelerate the antibiotic resistant crisis. The dilemma is whether the resistance in the food animal production give rise to further challenges in management of infectious disease in the clinical settings. Okay, if you see here, antibiotics are being used by human as well as the livestock, agriculture, fisheries, which can cause the development of antibiotic-resistant bacteria. Furthermore, the resistant bacteria from this domain can be spread to human either to direct contact, as in occupational exposure, or food consumption. And moreover, the antibiotic residues in the food can change the microbiome in the gut of human. So the resistant bacteria from both the domain ended up in the environment, which is a reservoir, and also harbor further resistant bacteria, and ends up it go back to human as well as livestock, agriculture, and fisheries. Antibiotic resistant bacteria from human further spread to other people to nosocomial transmission as well as community transmission. So as for today, moreover, due to significant linkages between the human health, animal health, environmental health, AMR has been recognized as a One Health issue, which necessitates a multidisciplinary, multi-sector, and coordinated approach to address the health threats. As for today, we'll be focusing on the environment related to livestock. We see avoparcin, vancomycin, chloramphenicol, nitrofuran, teicoplanin, norfloxacin are not for the use for livestock, food animal production industry. As for Malaysia, we adopted a phase out in banning antibiotics for 2019 colistin, and next effort were erythromycin, enrofloxacin, tetracycline, ceftiofur, tylosin, and phosphomycin. So EHRC collaborate with the DVS Selangor, which is a Department of Veterinary Services of Selangor, and we conducted to determine a study to determine a prevalence of AMR among bacteria in poultry farm environment, which involved 33 farms, 9 districts in Selangor. The sample was soil and effluent sample, which involved 511 isolate with gram positive 138 and gram negative 372. And here's the chart showing the number of isolate with the resistant rate with the antimicrobial tested based on the chicken production system, which is a broiler, blue color square, indigenous in orange square. Chicken housing system is a triangular in green color is a closed system, and a yellow triangle is open system. As you can see here over here, for zero resistance, which is a susceptibility, for isolate from the indigenous chicken production system shows higher prevalence compared to the broiler, which is in a blue color square. Whereas as it advanced for the resistant rate, broiler isolate from the broiler become higher than the indigenous. As for chicken housing system, the open system, which is in a yellow triangle, is higher than the green color, which is a closed system in general. So here's the resistance rate according to the antibiotics, gram-negative bacteria. If we see the higher resistance, which is more than 50%, can be observed through against cephaloxime as well as ampicillin, followed by gram-positive bacteria, which is fusidic acid, tetracycline, clindamycin, erythromycin, oxacillin, and benzylpenicillin. So our finding will become as what they call baseline study, and this was performed after the banning of tetracycline as well as erythromycin. Hence, our finding can be compared with a future study to look at the trend of the resistance following banning of those antibiotics. Okay, due to time constraint, we actually only focus on one bacteria, even though there are so many strains were recovered. 96 enterococcus were recovered from poultry environment. So enterococcus faecalis were 45 with Physium 35, others 16. Resistant of enterococcus become a cornerstone representing resistance among other gram-positive bacteria. Moreover, Enterococcus faecium was enumerated as one of the global pathogen list, which is known for escape pathogen. So this figure shows the antibiogram of Enterococcus isolate recovered from poultry environment in Selangor. Higher resistance rate can be seen against the tetracycline and erythromycin, whereas a moderate resistance can be seen over linezolid as well as high-level gentamicin. All isolates susceptible towards the tigecycline, and the rest will exhibit lower resistance. And we can see here the vancomycin is a lower resistant. So it can be corroborated with the banning of evaporin and vancomycin in animal feed in this country, since both antibiotics, which are similar in regard to chemical structure, were linked to VRE following its use in feed for livestock. Even other studies has reported of a reduction of vancomycin resistant rate following prohibition of its usage in the veterinary sector. So despite the low rate of vancomycin resistance, this finding is important and need to be scrutinized as well. Next will be the linezolid, is a moderate resistant. Linezolid is one of the last resort for the treatment of enterococcal infection among humans. And also linezolid was included in the WHO list of critically important antibiotics. And all the linezolid resistant enterococcus recovered from our study were multi-drug resistant. And 2 isolate of it exhibited linezolid resistant, vancomycin resistant enterococci. So relevant agencies should to investigate usage of linezolid in food production industry or any other antibiotics that can facilitate the development of LRE. And here are the further findings on gram-negative bacteria entitled multi-drug resistant gram-negative bacteria and extended spectrum beta-lactamase producing Klebsiella pneumonia from the poultry farm environment, and the URL is here. Points to ponder here for the gaps and limitation. As for our study, in a previous study, we did not quantify the antibiotic residues to correlate with the resistance level, and we focus only on environment. We did not determine the relatedness between the human as well as the poultry environment. And based on other reviews, majority of the research focused on identifying and quantifying discharge of various chemical and microbial pollutant, and there were limited data on the fate and also fate of the pollutant in terms of exposure and effect of the human towards the human. So considering the gaps and limitation, our current project is AMR in dairy farm under umbrella of tripartite program. So antimicrobial resistant and antibiotic residues in dairy farm. It involved 3 working projects. Working project 1 on prevalence of AMR or selected bacteria isolate among personal and milk in dairy farm associated with quantification of antibiotic residues. And next is working project 2 headed by a microbiologist, Madam Yunesuri, entitled Prevalence of AMR or Selected Bacterial Isolate in Dairy Farm Environment and Environmental Risk Assessment. Whereas working project 3, molecular characterization of AMR genes and linkage between human, animal and environment, headed by Dr. Shaidia. So for our future direction, One Health, One World concept, in a global concept, it integrates the epidemiological aspect that add to understanding the evolution of genetic relatedness of antibiotic resistant in pathogen or vectors, host as in human or animal, and the associated environment on a global scale. So in this context, livestock, the product which are imported and exported to and from different countries can be a source of global dissemination of antibiotic resistant bacteria. So in this scenario, collaboration between countries can take this concept initiative to control antibiotic resistance at global level. So here are my reference. Thank you. For further question and collaboration, can email to this. Over to moderator.
Thank you, Dr. Saksha, for the insightful presentation. We will now be moving to question and answer session. Please do not hesitate to send us your questions, and I will try my best to connect you to our distinguished speakers through the posted questions. Nevertheless, should the time does not allow us to address all the posted questions, our speakers will try to their best to answer the posted questions through their emails. It seems that we have questions. Let me have a look. We have one question for Dr. Asrah. How do you determine the antibiotics to be used in the study for drinking water study? Dr. Asrah, could you answer this question?
Okay. Okay, thank you, Sophia. So we determine the antibiotics for the drinking water study based on what we are using at the hospital, and also from what we have detected from the meat product. So because we would like to see what are the fit of antibiotics from both sources, from hospitals and also from the agriculture. So thank you.
Okay. And there's another question here. Because in your slide, they mentioned that you did mention about one of the isolates that resistant towards all the antibiotic tested. So do you know what is the isolate in specific?
Okay. So we have obtained one isolate that is resistant to all antibiotics, and we have identified it as E. coli.
So that's isolate is it E. coli?
Yes.
Okay. I hope that answers both the question to Dr. Asrah. So any more questions? Okay. I'm still waiting for more questions from the viewers. So you can type your questions because I'm looking through some of the questions here. Okay, one question to Dr. Saksha. It was mentioned selected bacterial isolates in current project of dairy farms. Which bacteria strains are you looking at? So do you have any specific things that you're looking at in your study, Dr. Saksha?
Yeah. We are looking at Staphylococcus aureus, Enterococcus species, as well as E. coli. We also screening through the remaining escape pathogen, which are the Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, as well as Salmonella. So back to moderator.
Okay, that's very direct answer. So you are looking at escape in general.
Yes.
Okay. One more question. What type of samples will be taking from human animal and okay. I think the question is what kind of samples that you taking from human, animals, and environment, like maybe swab samples or soil or anything. Maybe could you explain more on this to be specific for human, animal, and environment, Dr. Sasha?
Yes, yes, Miss Sophia. So as for human, we are taking nasal as well as hand sample, and for hand sample, we'll be taking pre and also post wash. In terms of we want to evaluate the effectiveness of the hand washing, and as for environment, we'll be taking soil and effluent water, and further we also taking milk for a food. For animal per se, we are collecting the cow dung. There is a research found the microorganism recovered from the rectal as well as the cow dung are equivalent. So we are collecting the fresh cow dung to link to the animal. So that's all.
Okay. Still waiting for questions. Any more questions to the viewers? Please type in your questions to the chat box. Our presenters are more than happy to answer your questions. Okay. All right, there's one more question. Also for Dr. Sasha. What type of molecular characterization will your team performing for dairy farm project?
Okay, Miss Sophia. So for this project, we'll be doing a whole genome sequencing. So we will select the bacteria with the high resistance following the susceptibility testing. So from each domain, which involve the animal, human, environment, as well as milk, and from each farm, for every visit, we have a total 3 visits. So for every visit, from here we can see the relatedness between all the domain, and also we can see the trend as well.
Okay. Thank you, Dr. Sasha. I'm still waiting for some questions, but if there is no other questions, I think we may end our question and answer session. Okay, we hope that this webinar has shed light on some research work that we are conducting at our institute and would spark interest and could initiate research collaboration with others. So I hope that today's session will benefit all the researchers that interested with this area of studies. And ladies and gentlemen, now we have come to the end of our webinar session. I would like to thank all participants for their support to make this session successful. You may subscribe to our National Institute of Health Malaysia YouTube channel and click for notifications whenever there is a new post. Before I conclude, I would like to announce that we will have the next session for IMR-SEAMEO webinar series on 20th of August 2022. So do mark your calendars. 28th of August 2022. The same time as today. And the theme for the next webinar is laboratory animals, rodents. We hope you can join us at this next session. Until then, thank you from all of us at IMR. Take care and stay safe. Bye-bye.